All right, so get this. We're talking about entire cities, but shrunk down to exist inside a single cell.
Wow.
You sent over this research on how cells manage this, and apparently it all comes down to liquid droplets.
Yeah.
Liquid droplets that just appear seemingly out of nowhere.
It does sound a bit like magic, doesn't it?
It really does.
These biomolecular condensates, as they're called, are like tiny, self-contained neighborhoods within the bustling metropolis of the cell.
And this research paper we're diving into today, it's like a blueprint explaining how these droplets form, grow, interact, and even break down.
Yeah.
It's all about understanding the physics at play, and it gets really wild.
Absolutely.
Yeah.
It all boils down to a delicate balance of forces between molecules, a constant push and pull of attraction and repulsion.
Okay.
The tiniest shift in energy, even something as small as a few kBt, that's a unit for measuring energy at the molecular level, can dramatically influence the formation of these droplets.
Okay, so instead of bulldozers and cranes, we're talking about these tiny forces dictating how these cellular cities get built.
Right.
But how does it actually work?
Yeah.
The research mentions phase separation as a key process.
Yeah.
What exactly is going on there?
So imagine mixing oil and water.
Okay.
They spontaneously separate, forming distinct layers, right?
Yeah.
Phase separation in cells operates on a similar principle.
Uh-huh.
It's driven by the inherent tendency of certain molecules to prefer sticking to their own kind rather than mixing with others.
So certain molecules are like, let's stick together, guys, and others are like, nope, we're heading over there.
Exactly. And the deciding factors in this molecular meetup are things like temperature, the concentration of different molecules, and the unique properties of each molecule involved.
Yeah.
It's like a complex dance, with each factor playing a part in determining who partners with whom.
But I'm thinking about all the incredible complexity of a cell.
Right.
Surely it's not just a simple two-way separation, like oil and water? Right. I mean, cells have so many different components, not just two.
That's a great point, and it's what makes this research so fascinating.
Yeah.
Cellular droplets aren't simple mixtures.
Right.
They're more like a multi-layered cocktail with a huge variety of molecules interacting in complex ways.
Okay, so we're moving beyond a simple two-ingredient mix to a whole molecular mixology situation.
Exactly.
But if there are so many ingredients, how do these droplets maintain their structure? It
Right.
can't just be about separating, right?
You're exactly right.
Okay.
And that brings us to another crucial aspect. The material properties of these droplets. Just like how different cocktails have different textures, think thick and syrupy versus light and bubbly, these droplets exhibit a range of properties too.
Right.
Some behave like simple liquids, while others are more viscoelastic, meaning they have both fluid and solid-like characteristics.
Viscoelastic. So they're not always just blobs of liquid sloshing around. They can be more structured.
Absolutely. And the cell actually takes advantage of this. Take centrosomes, for example.
Yeah.
Those essential structures involved in cell division.
Right.
They need to be fluid enough to grow rapidly during cell division, but also rigid enough to withstand the mechanical stresses of pulling chromosomes apart.
So these droplets are kind of like cellular chameleons, changing their properties to suit the task at hand.
Right.
That's incredible. But how does a cell actually control those shifts between a more fluid state and a more solid-like state?
It's an area of ongoing research.
Yeah.
But we know that the specific mix of molecules within a droplet plays a huge role.
Okay.
Think about it like adding different ingredients to your cocktail.
Yeah.
A dash of this, a pinch of that.
Right.
It can dramatically change the texture and behavior of the final product.
Right.
And just like a skilled mixologist, the cell can precisely tune the composition of these droplets to achieve the desired properties.
Okay, so we've got these droplets forming through a delicate balance of forces and molecular preferences.
Yes.
They can have different properties depending on their composition.
Right.
But cells aren't just empty spaces with floating droplets. There's all sorts of stuff in there.
Yeah.
Don't these droplets get stuck or lost in the chaos?
That's where the concept of surface tension comes in.
Oh?
Just like a soap bubble.
Uh-huh.
A droplet has a surface that interacts with its surroundings.
Okay.
And that surface tension plays a crucial role in the droplet's shape and how it navigates the cellular environment.
So surface tension helps those droplets maintain their shape and avoid dissolving back into the cell.
Right.
But I'm imagining cells being jam-packed like a busy city with all its infrastructure and people constantly moving around.
Yeah.
How do these droplets maneuver through that?
You're spot on. We're now stepping into the bustling city center of the cell.
Okay.
And navigating this crowded environment is another key challenge for droplets.
Yeah. Okay.
So Welcome back to our deep dive into the fascinating world of biomolecular condensates.
Right.
When we left off, we were picturing these droplets trying to navigate the crowded cellular environment. Right. It really is a bustling metropolis in there.
Right, like trying to hail a cab in Times Square during rush hour.
Yeah, exactly.
So how do these droplets avoid getting stuck in all that cellular traffic?
Okay, so that's where things get even more interesting.
Okay.
These droplets don't just passively exist in this crowded space.
Uh-huh.
They actually interact with their surroundings,
Okay.
bumping into filaments, membranes, and that intricate 3D mesh called the cytoskeleton.
Okay, so instead of just being pushed around by the crowd, they're actually engaging with it.
Exactly.
How does that work?
Well, it all comes down to a concept called wetting.
Wetting, okay.
Think about how a water droplet behaves on different surfaces.
Okay.
On a hydrophobic surface like wax paper,
Uh-huh.
it beads up, trying to minimize contact.
Right.
But on a hydrophilic surface like glass,
Yeah.
it spreads out, maximizing contact.
So it's like those droplets are choosing their dance partners based on who they have the best chemistry with.
Exactly, a perfect analogy.
Yeah.
And this wetting behavior plays a crucial role in how droplets position themselves within the cell.
Okay.
For example, a droplet might prefer to associate with a particular type of filament,
Okay.
using it like a highway to move around.
Right.
Or it might interact with a membrane,
Mm-hmm.
causing it to bend or even wrap around the droplet.
Whoa! So they can actually reshape their surroundings.
Yeah.
They're not just passive passengers in this cellular city, they're actively shaping it.
Right.
But you mentioned earlier that these droplets aren't static.
Right.
They're constantly changing, right?
Yeah.
What drives that dynamism?
That's another layer of complexity that makes this research so exciting.
Okay.
These droplets are far from stagnant blobs.
Yeah.
They're constantly being formed and reshaped by chemical reactions happening within the cell.
So now we're adding construction crews and demolition teams to our cellular city.
Yeah, exactly.
But hold on, I need a bit of clarification here.
Sure.
Are we talking about chemical reactions happening inside the droplet or reactions in the surrounding environment?
That's a great question.
Okay.
And it highlights a crucial distinction.
Okay.
The research describes two main types of active droplets.
Yeah.
Those that are externally maintained and those that are internally maintained.
Externally versus internally. Okay, walk me through this.
Okay, so think of externally maintained droplets like those bustling city markets.
Okay.
They rely on a constant influx of goods and resources from the surrounding area to maintain their activity.
Okay.
In this case, the goods are molecules that participate in chemical reactions occurring outside the droplet.
Right.
These reactions can fuel the droplet's growth and help it maintain a stable size,
Okay.
even as molecules are constantly moving in and out.
So it's like a city with a thriving import-export business.
Exactly.
But what about those internally maintained droplets?
Internally maintained droplets are more like self-sufficient factories.
Okay.
The reactions that drive their behavior happen within the droplet itself,
Right.
allowing them to potentially grow and even divide independently.
Yeah. Okay, that's really cool. It's like those off-the-grid communities that generate their own power and resources.
Yeah.
But if these internal reactions are driving growth, wouldn't those droplets just keep expanding indefinitely?
Right.
There's got to be some kind of control mechanism, right?
You're absolutely right.
Okay.
Uncontrolled growth would be disastrous for the cell.
Right.
And this is where those environmental interactions we talked about earlier come into play. For example, imagine a droplet growing and pushing against the cytoskeleton.
Okay.
The physical constraints of the surrounding environment can limit its expansion.
Ah, so it's like zoning regulations in a city, preventing skyscrapers from popping up everywhere.
Exactly.
But I'm also curious about those externally maintained droplets.
Okay.
You mentioned they rely on reactions happening outside.
Right?
How do they control their size if they're constantly exchanging molecules with their surroundings?
That's a really important point.
Okay.
Externally maintained droplets have a fascinating trick up their sleeve.
Okay, what's that?
They can actually suppress a process called Ostwald ripening.
Ostwald ripening. Okay.
Sounds a bit ominous.
Yeah, it does.
What exactly is that? Imagine a bunch of soap bubbles of different sizes.
Okay.
Over time, you'll notice that the smaller bubbles tend to shrink while the larger ones grow even bigger.
Right.
That's Ostwald ripening in action.
Okay.
It's driven by the tendency of molecules to move from areas of high surface curvature to areas of low surface curvature.
Okay.
In other words, those smaller droplets with their higher curvature are less stable and tend to lose molecules to the larger, more stable droplets.
Okay, so it's like those bigger companies gobbling up the smaller ones.
Exactly.
But how do these externally maintained droplets prevent that from happening?
It's all about that delicate balance of reactions we talked about.
Okay.
In these droplets, the rate of molecule production outside the droplet is balanced by the rate of molecule breakdown inside the droplet. This balance prevents those smaller droplets from disappearing and helps maintain a more uniform droplet size distribution.
So it's like those government regulations that promote fair competition and prevent monopolies.
Exactly.
It's amazing how these simple droplets are governed by such complex dynamics.
Yeah, it is.
But I'm also curious about those internally driven droplets.
Okay.
How do they even form in the first place if the reactions are all happening inside?
Right.
It seems like it would be hard to get things started.
That's a great question.
Yeah.
And it highlights another crucial aspect of droplet formation,
Okay.
the concept of nucleation.
Nucleation. Okay.
Think about how water freezes.
Right.
It doesn't just instantly turn into ice at 0 degrees Celsius.
Right.
It needs a starting point,
Okay.
a nucleus,
Yeah.
like a tiny dust particle or imperfection in the container to initiate the crystallization process.
So it's like that first domino that sets off the whole chain reaction.
Exactly.
But how does that translate to these internally maintained droplets?
Right.
What acts as their domino?
There are different theories, but one intriguing possibility is that specific molecules within the cell act as nucleation sites.
Oh.
For example, a particular enzyme might be anchored to a surface within the cell.
Okay.
This enzyme could catalyze the reactions needed to kickstart droplet formation, acting as a seed from which the droplet can grow.
Okay, so it's like having a dedicated team of city planners, making sure new development happens in the right place at the right time.
Exactly.
But once those internally maintained droplets form, how do they regulate their growth and division?
Right.
They can't just keep growing and dividing forever.
Exactly.
Okay.
And that's where things get even more intriguing.
Okay.
We're starting to uncover evidence that these droplets can communicate and interact with each other in fascinating ways, almost like a network of interconnected cities.
Wait, hold on. Droplets talking to each other?
Yes.
We need to dive into that?
Yeah.
But our listeners need a moment to process all this awesomeness.
You're right. We've covered a lot of ground already. Let's take a quick break, and when we come back, we'll explore how these incredible droplets interact and what that means for the cell as a whole.
Okay. I've had a moment to, catch my breath after all that incredible information about droplet formation and regulation.
Yeah.
Now, you were about to tell us about these droplets interacting with each other. It's like
Right.
they have their own social network inside the cell.
That's a fantastic way to think about it. We've been focused on individual droplets, but remember, a cell isn't just a container for one or two of these things.
Right.
We're talking about a whole bustling ecosystem with countless droplets interacting and influencing each other.
Right.
It's like upgrading our city analogy to a global scale.
So we've got droplets forming, dissolving, growing, dividing, bumping into each other. How does the cell orchestrate all of this without descending into total chaos?
That's the million-dollar question.
Yeah.
And it's where the real beauty of this system shines through.
Okay.
Remember how sensitive these droplets are to their environment? Well, the cell can actually exploit these sensitivities to fine-tune the entire droplet network.
Okay, so it's like the cell has its own control panel,
Right.
tweaking things like temperature, pH, and the concentration of different molecules to keep the droplets in check.
Exactly. And this level of control is crucial for maintaining order and efficiency within the cell.
Right.
Imagine a city without traffic lights or any kind of infrastructure.
Yeah.
It would be utter chaos.
Total chaos.
The cell, through its ability to manipulate the environment, essentially provides those traffic lights and road maps for its droplets.
But it's not just about preventing traffic jams, right? You mentioned these droplets might actually be communicating with each other.
That's right. They're not just bumping into each other randomly.
Right.
They're sending and receiving signals, exchanging information, and even cooperating to achieve specific tasks.
Whoa.
It's like those intricate communication networks that keep a city running smoothly.
So how are they sending these signals? Are we talking about tiny little cell phones or something?
Not quite cell phones, but the concept isn't that far off. One way these droplets communicate is through diffusion.
The diffusion, okay.
Remember how molecules are constantly moving around, driven by differences in concentration?
Right.
Well, those concentration gradients can extend beyond the boundaries of a single droplet, creating signaling pathways that influence the behavior of nearby droplets.
So it's like each droplet emitting its own unique aroma, and those aromas mixing and mingling,
Yeah.
creating a complex olfactory landscape that other droplets can sense and respond to.
What a fantastic analogy. And just like different scents evoke different responses,
Right.
these molecular signals can trigger a variety of droplet behaviors.
Okay.
Some droplets might be attracted to each other,
Uh-huh.
merging to form larger, more complex structures.
Right.
Others might repel each other, maintaining their distinct identities and functions.
So we've got droplets attracting, repelling, merging. It's starting to sound like a cellular soap opera.
I know, it's pretty dramatic.
But seriously, what are the implications of all this communication? How does it benefit the cell?
Well, one key benefit is increased efficiency.
Okay.
By communicating and cooperating, droplets can streamline complex processes, ensuring the right molecules are delivered to the right locations at the right time.
Right, okay.
It's like having a well-coordinated delivery system that ensures packages reach their destinations without getting lost.
Okay, that makes sense. And it also ties back to that idea of specialization we talked about earlier.
Right.
Different droplets with different compositions can perform different tasks,
Yeah.
just like specialized departments in a city government.
Exactly. And this specialization, combined with efficient communication, allows the cell to respond dynamically to changes in its environment.
Yeah, okay.
Imagine a city facing a sudden heat wave.
Right.
Different departments would spring into action.
Mm-hmm.
The Parks Department might open cooling centers.
Right.
The Transportation Department might adjust bus routes.
Yeah.
And the Water Department might monitor water usage. Similarly, the cell's droplet network can rapidly adapt to stressors or changing conditions, ensuring the cell's survival.
It's incredible to think about how much is happening at such a tiny scale.
Yeah.
And you mentioned earlier that this research is still in its early stages.
Right.
What are some of the big unanswered questions that scientists are grappling with?
One of the most fascinating areas of exploration is understanding how these droplet interactions contribute to the emergence of complexity within the cell.
Okay.
We know that individual droplets can exhibit complex behaviors,
Right.
but what happens when you have hundreds or thousands of them interacting?
Yeah, right.
Do new emergent properties arise? Can we predict or even control these emergent behaviors?
So it's like trying to predict the behavior of a flock of birds based on the behavior of a single bird.
Exactly.
The interactions create something entirely new and unpredictable.
Exactly. And that's what makes this research so exciting and challenging.
Right.
It's not just about understanding the parts. It's about understanding how those parts come together to create a functioning whole.
Yeah.
It's like trying to understand a symphony by studying individual notes.
And speaking of symphonies, I have to bring up that tantalizing idea you mentioned earlier, the possibility that these droplets played a role in the origin of life.
Right.
I know it's a huge topic, but could you give us a little taste of what that might have looked like?
Imagine the primordial soup.
Okay.
A chaotic mix of molecules constantly colliding and interacting.
Right.
Now imagine a few of those molecules purely by chance forming a primitive droplet,
Okay.
perhaps a simple membrane enclosing a collection of RNA molecules.
Right.
This droplet, through its internal reactions, is able to grow and divide, creating more droplets. Some of these droplets might, again by chance, develop the ability to interact with each other,
Right.
exchanging genetic information and forming rudimentary communities.
So it's like those first few notes of a symphony hinting at the incredible complexity that's to come. And over billions of years, these simple droplets, through countless iterations of variation and selection, could have evolved into the complex cells we see today.
It's a captivating hypothesis. And while there's still much we don't know, the study of biomolecular condensates is providing us with fascinating clues about how life might have emerged from non-living matter.
Well, on that mind-blowing note, I think we've reached the end of our deep dive.
Yeah.
You've taken us on an incredible journey through the bustling world inside ourselves.
It's been a pleasure.
And I'm left with a profound sense of awe at the complexity and beauty of life at its most fundamental level.
It's been a pleasure diving deep with you, and thank you, dear listener, for joining us on this exploration.
Absolutely.
Remember, there's always more to discover, so keep asking questions, keep exploring, and never stop being curious about the incredible world around us.
Couldn't have said it better myself.